Excitation contraction coupling, or EC coupling, is the phase that is just after excitation of the muscle but before contraction of the sarcomere begins. So what we see occurring during excitation contraction coupling is that the action potential that started near the neuromuscular junction spreads or propagates down the sarcolemma, and that spreading of the action potential stimulates the release of calcium from the sarcoplasmic reticulum, which is in close proximity to our sarcomere. So calcium then permits the myosin on the thick filament, or the thick myofilament inside the sarcomere, to bind to the actin on the thin filament, and that's when contraction begins. So EC coupling is essentially the physiological sequence of events that are occurring just after muscle excitation, but before the contraction of the muscle actually begins. So recall that the plasma membrane of the skeletal muscle cell had these small tunnel-like structures in gray here that are called the T-tubules, and on either side of the T-tubule is where the sarcoplasmic reticulum, or SR, butts up against the T-tubule. You'll see that the end of the sarcoplasmic reticulum that sits up against the T-tubule is expanded and flattened, and these expanded ends of the sarcoplasmic reticulum that come in close proximity to our T-tubules are called the terminal cisterns of the SR. So the T-tubules and the terminal cisterns of the SR are essentially located or situated just above the junction between the A band, which is the dark band in the center of the sarcomere, and the light bands, or the I bands of the sarcomere, that are at either side, or flank kind of either side of the Z disc there. So we see that the T-tubule in gray and the terminal cisterns of the SR are essentially in really close proximity to the A band, I band border within our sarcomere. So returning to this figure, this figure got a little bit small in the PowerPoint presentation I'm showing here. But what you're seeing here is that the action potential, these little purple arrows, are propagating away from the neuromuscular junction. So this is the neuron here that's forming our neuromuscular junction. So our action potential with these purple arrows here is propagating away from the neuromuscular junction. And we see that it spreads down the T-tubules when it arrives at a T-tubule, because the T-tubule is really just an infolding of the sarcolemma, or the plasma membrane of our skeletal muscle cell. So in the walls of the T-tubules, what we have are these small little voltage-sensitive proteins that are in close proximity to our terminal cisterns of our sarcoplasmic reticulum. And these small voltage-sensitive proteins are connected to calcium channels that sit between the T-tubule and the SR, on the terminal cisterns of the SR. So that way, when this action potential stimulates these voltage-sensitive proteins that's inside the walls of this T-tubule here, those voltage-sensitive proteins change shape. They open the calcium channels on the sarcoplasmic reticulum, and then the sarcoplasmic reticulum releases its calcium into the sarcomere that's deep underneath it. So if we take a look at what's happening after calcium is released into the sarcomere, we see that calcium binds to troponin on the thin myofilament. So recall that troponin was associated with a filament-like protein, or a thread-like protein called tropomyosin. And when the troponin changes shape after calcium binds to it, that moves tropomyosin out of the way, because initially tropomyosin was blocking the site on actin where myosin would bind to. So essentially, tropomyosin is kind of shielding myosin from binding to actin. When calcium is present, though, calcium binds to troponin changes shape, and that moves, when troponin changes shape, that moves tropomyosin along with it. And so when the muscle is contracting and calcium is present, myosin is then able to bind to the actin proteins on the thin myofilament and start to pull. And so this is where we start to see muscle contraction taking place here. But when the muscle is relaxed, myosin cannot bind to actin because tropomyosin is in the way. But when calcium is present, that calcium helps to change the shape of troponin, which moves tropomyosin, so that myosin can bind to actin and pull on the thin myofilament, which is going to cause our sarcomere to shorten. So after, when the muscle relaxes, the calcium release channels in the sarcoplasmic reticulum end up closing, and the calcium levels that are inside the sarcomere begin to drop, because what happens is that calcium is actively transported back into the sarcoplasmic reticulum. So when this calcium disappears, troponin reverts back to its shape during its resting state. Tropomyosin then blocks the ability of, or tropomyosin then blocks the myosin binding site on actin, so that the myosin heads can no longer grab onto the actin proteins on the thin myofilament.